All right. Good morning, everybody. Welcome to our Buckeye Neurotrauma lecture series. I'm excited to introduce today's speaker, Dr. Andrew Sass, who's MD, PhD, assistant professor in neurology. Andrew is also part of the chronic brain injury program, and his paper won chronic brain injury program paper of the year. That's why we've invited him here to give this Buckeye Neurotrauma lecture. He's doing some very interesting work with neutrophils and axon regeneration in the context of injury. Just a little bit about Andrew. He got his MD PhD from the Medical University of South Carolina. He did his residency in neurology at the University of Michigan, followed by a fellowship in traumatic brain injury and sports-related neurology. Let's see. He had an R25 fellowship as a fellow, and now holds a K08 grant. He has over 9 papers, including a Nature Immunology paper that's on this topic here, I think he'll tell you about today. And he's really working hard to establish in his own lab here at OSU, and also on the clinical side is working to build a concussion clinic at OSU as well. So Andrew's an impressive researcher, and we're really excited for him to share his work with all of you. So thank you, Andrew, and you can get rolling.
Thank you very much for the introduction, Dr. Godbout. All right, so I get the joy of telling you about my research today, and I'm going to talk to you guys about neutrophil-mediated axon regeneration. So I am required to give you a disclosures page. I have no significant disclosures. I am funded by the NIH with my K08, but I have no affiliations with any corporations or any consulting processes. So axon regeneration after trauma in the CNS is significantly compromised. And this is because of 2 things that occur simultaneously immediately after traumatic brain injury. There is both a loss of intrinsic mechanisms within the neurons themselves in adults that impede their ability to stimulate axon regeneration themselves. And then there's also a number of extrinsic factors that occur as a result of the injury that then contribute to impeding regrowth and regeneration of neurons after the trauma. And there are a wide array of them. They include excitotoxicity from other neurons through glutamate signaling, the activation of astrocytes and microglia, building a glial scar that is difficult to overcome. There's edema and swelling and breakdown of the blood-brain barrier and trophic support. And then there's also inflammation and activation of the immune system. So when we look at the inflammatory response after traumatic brain injury, it begins with the injury of the neuron itself. When the neurons are injured, they release damage-associated molecular pattern proteins, such as ATP, SB100, ACE, and HMGB. And these proteins are released into the extracellular space where they then bind to microglia and activate the inflammatory cascade. This then leads to the activation of neutrophils and monocytes and astrocytes, and then an infiltrate of peripheral immune cells migrates into the area, in addition to an activation of resident CNS cells, such as the microglia and the astrocytes. This process happens in a very time-dependent fashion. Immediately following the injury, we have the release of the DAMPs. We have that initial inflammatory cascade, which produces these pro-inflammatory cytokines that recruit in these peripheral immune cells. Within minutes of the injury, neutrophils are the first cells to migrate into this area, followed by monocytes over time. And then eventually, over days to weeks, we get lymphocytes that also migrate into the area. And so conventionally, when we think of neutrophils and their response to traumatic neuronal injury, the neutrophils come in, and as they extravasate into the area, the first thing they do is they degranulate and they release proteases and pro-inflammatory cytokines and reactive oxygen species. And the presence of these proteases and these reactive oxygen species breaks down the blood-brain barrier, makes it easier for monocytes to migrate into the area, also contributes to the recruitment of more neutrophils, more monocytes, more microglia, and then eventually the lymphocytes. And this is the first step in the cascade. Next, we have the monocytes that come in, and monocytes and macrophages come in response to the injury itself and being recruited in by the neutrophils. And when they first migrate into the area, they are thought of having a pro-inflammatory activation state in which they also act similarly to neutrophils, where they produce more interleukin-1, a pro-inflammatory cytokine, so they can then recruit more cells into the space, produce TNF-alpha, which can be neurotoxic and injure neurons directly. They also produce chemokines such as CCL2 that then also recruit more cells in. And this kind of, you can imagine how this perpetuates a pro-inflammatory process. The interesting thing about monocytes and macrophages is that they're believed to be a key cell that makes the switch, at least conventionally after trauma, from the pro-inflammatory phase to the immune resolution phase or the anti-inflammatory phase. And so at some point, there's a switch in the macrophages from producing more pro-inflammatory cytokines to then producing anti-inflammatory cytokines that then inhibit this process and feed back to kind of calm down this inflammatory response. But this is, this has always been monocytes, and this pathway, this conventional pathway of neutrophils come in and do injury, and then monocytes come in and do injury, and then eventually they switch, has kind of been in the literature for a long period of time. So we wanted to investigate this more mechanistically as to how these cells are involved in this process. And to do this, we used some very simple models of axon trauma. So the model that we use primarily is the optic nerve crush model. This is a great model for injury. And the reason is because your brain has 100 billion neurons in it, and they are going in many different pathways. But in the optic nerve, all of your neurons are lined in the retina itself. And then all of the axons come together in one bundle down the optic nerve, and they all signal down the optic nerve in one direction. Retinal ganglion cells from the retina are derived from the CNS, and so they are part of the central nervous system. It is an isolated long fiber tract that we can isolate and manipulate and then do mechanistic work with. And it's a simple model to look at mechanistic studies compared to the complexity of whole brain traumatic brain injury. And the other thing that is very unique about the optic nerve crush injury is that it is a model in which inflammation can actually promote axon regeneration. And so this is different than other models of injury, such as spinal cord injury and more conventional models of traumatic brain injury. In this model, we can do these we can do this surgery to expose the optic nerve and do a micro dissection and then mechanically crush the nerve to create the trauma. This is representative of a severe injury to the neurons. We can then inject factors into the eye directly, sterile particulates that will then recruit in immune cells, creating a sterile inflammatory response where these immune cells can then interact with the retinal ganglion cells. Over a period of weeks, the interaction between the immune cells and the RGCs then stimulates axon regeneration across that crush injury, and those axons start to regenerate. And this, as I said, this is quite unique. And so what does this look like? And so the sterile inflammation that we use is the Zymosin model. Zymosin is a fungal wall extract. And this comes from a previous paper by a previous postdoc in Dr. Stahl's lab, in which they looked at how Zymosin is the unique particulate that is able to stimulate sterile inflammatory axon regeneration. And so starting here in the top left, what you see is longitudinal sections of optic nerves. And so on the left over here, this is where the eyeball would be. The star denotes the crush injury. And then these optic nerves were stained with GAP43 or growth-associated protein 43 antibodies to label regenerating axons from the crush injury. The other thing that's really nice about the optic nerve crush injury is that you can overlay a grid on the nerve where you can measure out distances from the crush site to see how far axons regenerate down the optic nerve. And what we found was that the Zymosin injected into the eye stimulates axon regeneration quite robustly. But if we use LPS, a bacterial protein, you find that LPS is not able to stimulate axon regeneration. And if you just inject PBS into the eye, the presence of just injecting solution into the eyeball does not stimulate regeneration on its own. And down here, we were able to quantify this and look at the amounts of regeneration. So the first question I asked is, well, what are the cells that respond to the presence of the Zymosin and this axon regeneration? And when we look at the infiltrating kinetics of the cells migrating in over time, we found that the predominant cell migrating into the eye early and at the highest numbers is the neutrophils. And we found that very interesting based on the background literature that I described to you about how we think of neutrophils as detrimental. And so we asked the question, well, what role do the neutrophils have after the Zymosin injection? Are they helpful? Are they detrimental? Is it really those monocytes and macrophages that we saw and the neutrophils are getting in their way? And so we wanted to inhibit neutrophil infiltration. And so to do this, you guys need to know that neutrophils conventionally migrate into tissue through the chemokine signaling of CXCR2. And so we developed an anti-sera against CXCR2 to inhibit neutrophil migration. And we took mice and we split them into two groups. One group got a control antibody and the other group got the anti-CXCR2 antibody. And both groups were subjected to the optic nerve crush and both groups got Zymosin injected intraocularly. And then we treated the mice every 2 days with the anti-CXCR2 or the control sera and waited 2 weeks to harvest the nerves and look at axon regeneration. And when we did this, we were quite surprised by the amount of regeneration that occurred. And so what you can see here on your right is, once again, representative images of the optic nerves. This time, instead of staining with GAP43, these are clarity images in which we injected a tracer that is taken up by the regenerating axons. But the eye would be here. The star, once again, denotes where the crush injury is. And then axons grow longitudinally down. And so when we did the crush injury and just injected PBS, there was very little regeneration. When we injected Zymosin, we got a significant amount of regeneration within 2 weeks. If we injected PBS and gave the anti-CXCR2, we didn't get any significant regeneration. But the Zymosin plus the anti-CXCR2 produced far better regeneration than the Zymosin alone. And we quantified this here on the left, where we looked at the amount of neurons that were regenerating at the distances past the crush injury. We also looked at the viability of the retinal ganglion cells in the eye itself. And when you do the crush injury and inject PBS, we have a significant dropout of retinal ganglion cells. This is staining of retinal ganglion cells with beta tubulin and BRN3A markers. You can see what a naive retina looks like for a comparison of how many retinal ganglion cells you can see without any injury. And what we did was with the PBS, we saw that there was a significant dropout in the number of surviving axons. When we gave the Zymosin, there was more, but with the Zymosin and the anti-CXCR2, we had the most surviving axons that were then able to regenerate. So then the obvious question after we saw that there was better regeneration was, were we able to block neutrophils? And what we did when we looked at the kinetics of the infiltrate over time, looking at 1, 3, 5, 7, and 10 days after the injury, we found that we initially blocked neutrophils from migrating in. So on day 1, in our anti-CXCR2 treated group, we didn't have any neutrophils migrate in compared to the Zymosin with the control sera. But the neutrophils eventually found their way in much later in the time course, still got to similar levels. And so I found that quite odd that, yes, we blocked the initial process, but they still managed to get in. So we then started to ask questions, well, what are these neutrophils? Are they different than what we would expect? And so we began with some phenotypic descriptions of the neutrophils. And what we found was that if you give the Zymosin and the control sera and you look at the neutrophils, they look like conventional neutrophils. They are multi-segmented, hyperlobulated neutrophils. They have granules down here that you can see with the red arrows. But in the neutrophils that we isolated out of the eyes from the mice treated with the anti-CXCR2, our neutrophils had this ring-formed nuclei. They still had granules, but they were shaped differently. These are considered an immature-looking neutrophil when you look at the maturation process of the neutrophil. And so they appeared visually less mature. We did some very extensive flow cytometry to look at these neutrophils and the markers and characteristics of them between the two groups. And what we found was that in addition to them having this ring-formed nuclei, they also had a lower expression of Ly6G, which is a protein specific for neutrophils. So they had lower expression of Ly6G. They had higher expression of CD14, which is actually a marker that we conventionally think of being found on monocytes and macrophages. And they had lower expression of Ly6B, which is another neutrophil marker. Then finally, there is a marker CD101, which is specific for mature neutrophils. So neutrophils that express CD101 are considered mature and circulating, and neutrophils that are CD101 negative, that are Ly6G positive but CD101 negative, are immature. And we found that in our mice that were treated with the anti-CXCR2 after the crush injury, there were less mature CD101 positive cells in those eyes. So after we did this flow cytometry, we then moved on to do some molecular characterization, doing real-time PCR to look at transcripts of different markers. And to borrow from the monocyte macrophage literature of that M2 macrophage that's anti-inflammatory, we looked at markers of arginase, interleukin-4 receptor alpha, and mannose receptor, which are markers of alternative activation and anti-inflammatory markers. And what we found was that our Zymosin neutrophils that came from the mice treated with the anti-CXCR2 had much higher expression of arginase, IL-4 receptor, and mannose receptor compared to the neutrophils that were found in the Zymosin eyes treated with control sera, indicating some alternative activation factors associated with it. So people might ask the question, well, are these neutrophils actually monocytes? Because I showed you data that they have lower expression of Ly6G, they appear immature, they had some markers that we see more conventionally on monocytes and macrophages. So here's our flow plot, and these are our neutrophils here by Ly6G, and these are the monocytes that are Ly6G negative. And when we look at the monocytes, we find that although our anti-CXCR2 neutrophils are lower expression of Ly6G, the monocytes are negative for Ly6G. The monocytes in green here express the macrophage colony stimulating factor receptor, which is specific to monocytes. And the monocytes also had lower expression of myeloperoxidase, which is a reactive oxygen species generally seen in neutrophils, not in monocytes. And so we did not, based on this flow characterization, we did not think that this was some sort of alternative monocyte macrophage, but it was a neutrophil. And so the next step was to get a deeper dive into the transcriptomics and the description of these neutrophils. And what we did was we flow sorted these neutrophils, isolated, purified cultures of them, and then submitted them for single cell RNA-seq, where we submitted 10,000 neutrophils from either mice that were treated with the control sera after getting the optic nerve crush, or mice that were treated with the anti-CXCR2. And when we looked at the RNA, the transcriptomics, and broke them down into a UMAP plot, we found that within each group, the neutrophils were actually heterogeneous, regardless of the treatment they received. They broke down into 3 different clusters: cluster 1, cluster 2, and cluster 3. But when you just look at this, you'll see that there is less of cluster 1 in our anti-CXCR2 treated group and more of cluster 3. And we began to ask questions about what is the maturity state and the activation state of these different clusters. And so beginning with looking at markers of neutrophil maturation, we looked at cluster 1, cluster 2, and cluster 3. And I'm going to focus on cluster 1 and cluster 3 because what we found was that cluster 1 expressed all of the mature factors. So they had high expression of Ly6G and CXCR2, S100A8, and that CD101 marker. So those were all found in high expression in cluster 1. And in cluster 3, we found low expression of those. But when we look at immature markers of immature neutrophils, such as these transcription factors EF2, CDK1, CDK4, we found that they had much higher expression in this cluster 3. And it turns out that although I don't have cluster 2 in this plot, cluster 2 is kind of a transition state. And so based on maturation markers, you can think that cluster 3 is immature, cluster 2 is a transition state, and then cluster 1 is the most mature of the groups. Additionally, we looked at markers of conventional inflammation or inflammatory activation versus alternative activation. And so when we look at markers such as TNF-alpha, interleukin-1, FAS, ICAM, markers that are pro-inflammatory, we found that they had the highest expression in cluster 1 in either treated group. Whereas if we look at those markers of alternative activation that we think of with monocytes, markers such as TGF-beta, arginase, mannose receptor, growth factors such as insulin-like growth factor, we find that they have much higher expression in cluster 3. And so not only is cluster 3 more immature, but it also appears to be alternatively activated in an anti-inflammatory state. And we can kind of, to delve into this a little bit more, instead of just looking at this from a heat map, we can actually overlay the gene expression onto the clusters themselves to see what that actually looks like. And so if we take some markers of neutrophil maturation, such as S100A8 or CXCR2, you can find that this heat map shows that the highest expression of these proteins is found in cluster 1, and the lowest expression of these proteins is found in cluster 3. Whereas alternatively, those markers arginase and mannose receptor are found almost exclusively in that cluster 3 population. And so that kind of gave us an idea of what these subsets were and within the heterogeneity of these neutrophils. And so far, what I think I've shown you is that in the optic nerve crush model with Zymosin-induced axon regeneration, the neutrophils are the predominant cell that infiltrated into the eye. That blocking neutrophil chemotaxis with a CXCR2 anti-sera delayed the infiltrate of neutrophils and enhanced axon regeneration. And that the neutrophils that did find their way into the eye after the crush injury and inhibition with the anti-CXCR2 had a unique phenotype not seen with conventional neutrophils, in which they had lower expression of Ly6G, they were CD14 positive, they were immature and CD101 negative, they had that ring nuclei. And then they had a unique transcriptome that was defined by immature and alternative activating factors. But all of this that I've shown you so far is just a description of what's going on. And so we really wanted to know, well, do the neutrophils have a beneficial role? Can these alternatively activated neutrophils have a beneficial role? But this is difficult to do, to take neutrophils out of the eye and then get enough of them to transfer them to into other animals and do a significant amount of work with. But what we found was that you can take Zymosin and you can inject it intraperitoneally. And if you just wait a couple hours, the presence of the Zymosin in the peritoneal space, if you do a lavage, you'll get lots of conventional Ly6G high neutrophils out of the peritoneal lavage. If you wait 3 days though, and you do a lavage over 3 days later, you actually get these Ly6G low neutrophils. And so when we do this by giving the Zymosin IP and then lavaging it and isolating these neutrophils, we can then collect millions of neutrophils that we can then use for all kinds of adoptive transfer experiments to look at how they're actually functioning on their own in the absence of the Zymosin. And so the first part of this is to show you that these Ly6G low neutrophils that we get out of the peritoneal lavage is similar to the ones that we get out of the eye. And so we did flow cytometry, and once again, we get lower expression of Ly6G. The red are the 3-day neutrophils, the blue are the 4-hour neutrophils. We get higher expression of CD14 out of these neutrophils. When we do PCR to look at those markers of alternative activation, the neutrophils on day 3 have higher expression of arginase and IL-4 receptor and mannose receptor. And then what we did was we took them and we subjected mice to the optic nerve crush. And then instead of injecting Zymosin in the eye, we took these purified neutrophils and directly injected them into the eye. And what we found was that when you directly inject these Zymosin stimulated neutrophils that are 3 days old, that are have Ly6G low and alternative activating factors, they're able to stimulate significant amounts of axon regeneration on their own in the absence of the Zymosin in the eye. And they are able to stimulate that more than 4-hour mature neutrophils or bone marrow neutrophils that would be a different type of immature neutrophil, or just the PBS injection alone. So the next question that we asked was, well, do neutrophils need monocytes in order to stimulate this axon regeneration? And so to do this, we took those IP neutrophils and injected them into either wild type mice or CCR2 knockout mice. CCR2 is the chemokine receptor that monocytes conventionally use to migrate into the eye. And when we inject the neutrophils into the eye and then wait a day and then harvest the vitreal fluid to look at the cells, in a wild type mouse, when we inject neutrophils, we get neutrophils in the eye, but we also find monocytes. When we inject neutrophils into the CCR2 knockout mouse, we get neutrophils, but we don't get an infiltration of monocytes into the eye. And then we could look at axon regeneration in both the wild type mouse and the CCR2 knockout mouse. And what you can see is very similar to the past. The eye would be here. The crush injury is denoted by the line. And you can see with this gap 43 staining that There is similar amounts of axon regeneration with the adaptive transfer of the neutrophils, even in the absence of monocytes themselves. And so in this set of experiments, we found that the Ly6G low neutrophils were able to be produced by Zymosin outside of the optic nerve crush model, that we can adaptively transfer them into another mouse that has been subjected to optic nerve crush and stimulate axon regeneration. And the effects of these neutrophils can be done without the presence of monocytes that are thought to be, in a lot of literature of trauma, thought to be important for that stimulation of axon regeneration. And so the neutrophils clearly have a direct effect on the neurons. And so the next steps were to look at how the neutrophils interact with the neurons and how they're able to stimulate this axon regeneration. And to do this, we moved out of the mouse model and into in vitro assays to further look at this mechanistically. And so we can take mice and we can take the eye and isolate the retinal ganglion cells, and we can dissociate them into low density cultures, and we can plate them so that we can grow them individually. We can also inject Zymosin intraperitoneally. We can get the cells, we can purify out the neutrophils, and then we can put the neutrophils into culture with these RGCs. We can let them incubate together and then look at how they stimulate or affect neuron viability and axon regeneration. And so we did this. And so if we take the retinal ganglion cells out of the mouse and we just plate them in culture and we don't do anything to them, they grow this little tiny neurite that kind of grows out. And we can measure this neurite with computer programming. If you give them a growth factor, their growth factor of choice is ciliary neurotrophic factor, CNTF. They grow these very long neurites. If we put the Zymosin directly into the culture without any cells, they don't stimulate any axon or neurite outgrowth. But when we took those Zymosin stimulated neutrophils and placed them into culture, we found that they were able to grow these long neurites out again that we quantified over here. And then the next question we asked was, well, are these neutrophils generating this effect by directly interacting with the neurons, or are they secreting some sort of factor, soluble factor into the protein or into the media? And so to do this, we then took neutrophils and we put them into media, let them secrete factors for 24 hours, then removed the neutrophils out of the media and had just this neutrophil conditioned media, and placed that neutrophil conditioned media then into the culture with the neurons. We also did things like heat shock that conditioned media to dissociate the proteins or filter the proteins. And we found that the neutrophil conditioned media alone was able to stimulate neurite outgrowth. But if you heat shock the protein or heat shock the media before adding it to the culture, it does not stimulate neurite outgrowth, indicating that the neutrophils are secreting a soluble factor. So we then did a growth factor array, and we did a number of different types of growth factor arrays to look at what factors might be found in that media that are being secreted by the neutrophils. And so we have untreated media versus neutrophil conditioned media, and found that neutrophils were secreting a number of growth factors that are beneficial to neurons. And that included nerve growth factor beta, insulin-like growth factor, TGF beta, platelet-derived growth factor, stem cell factor. And so all these factors were being produced. We then did some PCR of the factors, and we started with the most prevalent ones, the NGF and the IGF. And so we took those Zymosin stimulated neutrophils, either the mature 4-hour ones or those immature 3-day neutrophils, and looked at NGF and IGF-1 and found that at the transcript level, these 3-day neutrophils were transcribing NGF and IGF-1. We also did ELISAs to look at that conditioned media and found that there was higher levels of NGF and IGF-1 in the 3-day neutrophil conditioned media compared to 4-hour neutrophils. We also went back into our optic nerve crush mouse model and took vitreal fluid from the mice that were subjected to the optic nerve crush and got either that control sera or that anti-CXCR2, and found that mice that got the anti-CXCR2 and Zymosin had high levels of NGF in the vitreal fluid and high levels of IGF-1 that weren't found in our control groups. And then finally, just to verify that those neutrophils were also able to directly stimulate outgrowth, we did do a series of experiments in which we did the optic nerve crush, We then injected the Zymosin and treated the mice with either the anti-CXCR2 or the control sera, and then took the neutrophils out of the eye and then put them into media with the retinal ganglion cell cultures, and found that the neutrophils taken out of the eye and placed into the neuron cultures were they themselves able to stimulate neurite outgrowth. So the next step was to determine if these growth factors that were being produced were the growth factors that are responsible for the neurite outgrowth. And so we then did a series of inhibition assays to block NGF and IGF-1 in these neurite outgrowth cultures. And so we have our retinal ganglion cells, we have our neutrophil conditioned media. We then treated the media with an anti-IGF-1 antibody, an anti-NGF antibody, or the two inhibiting antibodies together, or an isotype control antibody to see if there was a non-specific antibody effect. And what we found was that when you inhibit either IGF-1 or NGF, you have significantly decreased regeneration compared to the no antibody treatment or the isotype control. And then if you inhibit both of them, then they have the least amount of regeneration or neurite outgrowth in vitro. And so then we did the inverse of this, which is ask the question, how well does NGF and IGF-1 stimulate neurite outgrowth if they are responsible for this? And so we took recombinant NGF and recombinant IGF-1, and we then subjected mice to optic nerve crush and then injected the IGF-1 or the NGF directly into the eye, or the combination of the two growth factors together, and then quantified the amount of axon regeneration. And here you can see it on the right that, you know, the eye would be here, the line denotes what the crush is. And the GAP43 staining shows that IGF-1 stimulates some regeneration. NGF does a little bit more, but the combination of the two growth factors together clearly drives the most amount of regeneration. So now we wanted to look at this specifically with inhibiting the NGF and IGF-1 in the mouse model. And so we injected the mice. We started by injecting the mice with the Zymosin intraperitoneally. We harvested those Ly6G low neutrophils. We then mixed them into solution with a neutralizing antibody against NGF, IGF-1, or the combination two antibodies. And then we injected either the neutrophils or the neutrophils with the neutralizing antibodies into the eyes of mice that have been subjected to crush injury. And when we did this, what we found was that if you do the crush injury and then put the neutrophils in with the control antibody that does not neutralize IGF or NGF, then you still get significant regeneration down the length of the neuron. If you inhibit IGF-1, you get less regeneration. This is seen here in the blue bar. If you inhibit NGF in the presence of the neutrophils, you get less regeneration also. And then if you inhibit both growth factors, you get significantly less regeneration, almost back to the level if we just gave no cells at all after the injury. We also did RGC viability on the whole mouse to look at how many cells lived after inhibition of the neurite outgrowth, or sorry, after inhibition of the growth factors that are being produced by these neutrophils. And if you, on the right, you can see this BRN3A staining. And this quantification, we found that if you give the isotype antibodies that don't inhibit the growth factors, that you still have a significant amount of viable cells that are protected by the neutrophils. If you inhibit just IGF-1, there's a decrease, but you get the fewest surviving RGCs if you inhibit both the NGF and the IGF-1. And so from all of this work looking at these growth factors and these addition where we added in growth factors and then removed growth factors and inhibited them, we found that the mechanism of this neutrophil mediated axon regeneration in this mouse model was at least partially mediated by the secretion of growth factors, including NGF and IGF-1. I showed you data that IGF-1 and NGF, or the combination of the two, can stimulate axon regeneration on its own, and that when we inhibit these growth factors, that it prevents axon regeneration and inhibits neuroprotection that occurs by the presence of these immature neutrophils. So once we did this in the optic nerve crush injury model, the obvious question is this specific to just the optic nerve crush model, or does this apply to other models of neuronal injury? And so we moved from the optic nerve crush model into the spinal cord injury model. And so we began with some in vitro assays. Instead of using the retinal ganglion cell dissociated cultures, we started using dorsal root ganglion neuronal cultures from the spinal cord to look at how neutrophils interacted with the DRGs. And so in the same way that we did this with the RGCs, we plated just DRGs. And if you plate a DRG and you don't give it any growth factors or anything to stimulate it, it has very little neurite outgrowth. We used a model of conditioned injury as our positive control, in which we prime the neuron for neurite outgrowth with an injury prior to plating it. But this produces a strong positive control in the literature of strong neurite outgrowth after injury. If we put zymosin into this culture without any other stimulus, the zymosin itself did not stimulate neurite outgrowth. We tried bone marrow neutrophils, and they had a little bit of neurite outgrowth. We then added those zymosin-stimulated Ly6G low neutrophils that had more neurite outgrowth, significant longer neurites that looked similar to the condition injury. We found that if we added the neutrophil conditioned media, we once again got that good neurite outgrowth. And if we heat shocked that media, once again, we recapitulated the same effect that we saw with the retinal ganglion neurons. And all of this is quantified here. So then we moved into the spinal cord injury model, so the dorsal spinal cord injury. And so for this injury, we took mice and they were subjected to a dorsal column lesion at T8. Some mice prior to the dorsal column lesion at T8 were subjected to a conditioning injury as our positive control to stimulate that axon regeneration at the lesion site. And then the other mice that did not receive the sciatic nerve crush injury as their conditioning lesion, on the day that they got the T8 lesion, they also got an injection of neutrophils into the sciatic nerve at the site as close as possible to the DRG. And they got either bone marrow neutrophils or those Ly6G low zymosin stimulated neutrophils. We then waited 7 weeks and injected tracer to highlight those axons. And then at 8 weeks, we then harvested tissues. And what we found was that if you do the T8 transection and you just inject PBS, here the star denotes where the transection occurred in each of these, and here these are the tracer labeled axons that grow up to the injury, but they don't grow across the injury. If we injected the bone marrow neutrophils into the sciatic nerve after the crush injury, they grow a bit farther, but still don't transect across or cross the lesion. But with the neutrophils, the 3-day neutrophils that we injected, we found that those neutrophils were able to cross the T8 lesion and extend with axon regeneration forward past the injury. And so we quantified this looking at either retraction from the site, measuring backwards to where the longest neuron was measured, or as regeneration if it was past the neuron. And so that is quantified here, where we found that the 3-day neutrophils were able to significantly stimulate regeneration instead of retraction compared to the other conditions. All right. So we, in this part, working with the dorsal root ganglion cells and with spinal cord injury, we showed that Ly6G low neutrophils and their secreted factors can stimulate neurite outgrowth in the DRGs. And so this isn't just specific to retinal ganglion cells. Also showed that adoptive transfer of these Ly6G low neutrophils can stimulate the DRG axons to grow across the transected glial scar in the model of T8 dorsal lesion injury for spinal cord injury. So the final steps of this project and this paper were to then ask the question, is this translatable to humans? And to look at human neutrophils. And so we began this process by starting with HL60 cells. So HL60 cells are a acute myeloid leukemia cell line. They are An immature neutrophil, they are used in the literature where you can place them into culture and you can add retinoic acid to stimulate them to an immature neutrophil phenotype and generate a immature neutrophil in vitro. We also used some other control cells, some DG75 cells, which are a B-cell lymphoma, so a different type of immune cell. And the first thing we did was look and see if these human cells produced any growth factors. And we did this by putting them into media and then taking the supernatant off the media and looking at the amount of NGF present in the media, and found that these HL60 cells were able to produce measurable amounts of NGF. We also, after culture, looked at a transcript of the marker arginase, which is that marker of alternative activation, and found that these immature neutrophils human neutrophils were able to have high expression of arginase and transcript of the NGF. We then took the HL60 cells and we put them into culture with the retinal ganglion cells and looked at the amount of neurite outgrowth that they did. And similar to the work we did with the mouse neutrophils, we created HL60 conditioned media. And we found that the addition of the HL60s or their conditioned media was able to stimulate significant longer neurite outgrowth compared to the RGCs alone or the DG75 B cell line. And because we identified NGF as a growth factor that was being produced by these HL60s, we then did this similar inhibition assay in which we took the HL60 cells and placed them into culture with the RGCs and then added either an isotype control antibody or an anti-NGF antibody to the conditioned media, or heat shocked it. And we found that by inhibiting NGF with that neutralizing antibody, we had decreased amount of neurite outgrowth compared to the control antibody. And if we heat shock that media, we once again completely abrogate any effects of the factors that are produced by these HL60 cells. And then the last thing we did with these HL60 cells is that we took the HL60s and the DG75s, or those 3-day IP Zymosin neutrophils, and we injected them into SCID mice, immunosuppressed mice that won't reject the human cells, and looked at the amount of axon regeneration. And we found that if you take those HL60 cells and inject them into the eye, and we look at the amount of regeneration, we can see that the HL60s have similar amounts of neurite outgrowth and axon regeneration to what we see with those 3-day Zymosin stimulated neutrophils. But the DG75s, that B cell line, does not have the ability to stimulate neurite outgrowth. And we quantify that over here. So in summary of all of the work that was done in this paper, we identified a unique subset of pro-regenerative neutrophils that are Ly6G low, CD14 positive, CD101 negative, and arginase, IL-4 receptor, and mannose receptor positive. We found that they have a distinct transcriptome that separates them from mature neutrophils when we did single-cell RNA-seq. These immature alternatively activated neutrophils can stimulate RGC axons and DRG axons to regenerate after acute traumatic injury, and that the mechanism in which these neutrophils stimulate regeneration is through the production of growth factors that can directly stimulate these axons. So from this work, we have a lot of work moving forward that's very exciting in the lab. And so although I showed that the neutrophils can directly do this effect, we know that in the Zymosin model, there is the recruitment of monocytes, and we are looking at how the interplay between the neutrophils and the monocytes might be synergistic in order to produce the best regeneration. One of the important questions from my K grant that I'm exploring right now is, how do these regenerative neutrophils get to the site of neuronal injury? Is there a way that we can selectively enrich them? So we inhibited CXCR2, but so they must get there through an alternative pathway. What is that? Can we make it more likely to get pro-regenerative neutrophils? And then, although we did some work with an HL60 cell line that is similar to an immature neutrophil, we are doing work looking for a true human correlate of an immature or pro-regenerative neutrophil in patients who have suffered acute neuronal injury. And so thank you for listening to my talk and learning about the paper and all the work that we do in our lab today. I greatly appreciate it. I need to give all of the support and acknowledgments to my mentor, Dr. Segal. He's the chair of the Department of Neurology. I'd also like to thank Dr. Godbout and Kedar for supporting me with the CBI program. And none of this work could get done without the people in the lab. And so I'd like to thank my technician, Alex, for all of the hard work he does, and all of the people in Dr. Segal's lab who have helped me and supported me through all of this and all of the things that they do. And finally, my funding. And so thank you for listening. I'm happy to answer any questions that you guys have.
Okay, let's see if we have any questions. I think, the way that we like to do it is to ask questions live if we can, or you can post in the Q&A. So Andrew, I have a question about, so do you see this reparative neutrophil population, is that same profile in the bone marrow and in the blood, or does that profile sort of develop as it hits the tissue?
Yeah, so that's a really great question. So one of the things that we did do that I've done since this paper went out is we did a comparative transcriptomics of bone marrow neutrophils, circulating neutrophils, and the neutrophils at the site of injury after the Zymosin injection, and found that although they have the immature factors in the bone marrow, so they have Ly6G low, they're CD101 negative, and that is held up in circulation. All of those alternative activating factors and the transcriptomics associated with those growth factors, that does not occur until they get to the site of injury. And so although they have the immature phenotype, their activation state and that pro-regenerative state really occurs through some sort of on-site signaling pathway that we have yet to specifically elucidate. And
is that all these found specifically in the epicenter of that injury, or is it that you can find more distal to the injury? You think, I guess what I'm trying to say, is there a temporal, spatial?
Yeah, so the thing about I guess that would be a hard thing to do. I can't answer that directly because we haven't done that experiment looking at it spatially, but if you look at the retina, you do a retinal whole mount and look at RGC dropout. The dropout occurs in the entire retina after the crush injury. So you get the Wallerian degeneration and you get the RGC dropout, and it happens everywhere with the model because of the severity of the injury. And so you get immune cells in the whole retina area. But there's a separate question, which is there is the interaction of the immune cells at the retina, but there's also what's going on at the crush injury itself, because we know through all of the spinal cord literature that there has to be a permissive environment at the crush injury in order to allow those axons to generate across the glial scar. And so we know that small amounts of the cells migrate there, but I have not explored their role in interacting with microglia and astrocytes at the crush injury yet.
Okay. Well, looks like we have somebody with a raised hand. Kedar, can we get Pilar to ask live?
He just actually answered my question. I was going to ask whether he had looked at how the neutrophils affect the glia, like the microglia and the astrocytes in the retina, but he just said that he didn't.
Yeah.
Oh, okay.
Yeah. So along those lines, so as part of this paper, one of our reviewers did ask us about that. So we did do some time course looking at glial activation, and we found that there was a significant increase in glial activation within 1 day. So high expression of GFAP positive Mueller cells, which are the astrocytes in the eye, and microglia. With the adoptive transfer of the neutrophils, that activation state of the glial cells died down within 7 days and tended to disappear by 14 days completely. So we know that there is an interaction, and we know, although my lab doesn't look at glial cells, we know that they also have an important role in the trophic support and the axon regeneration process.
Cool. Did you see it through PCR? like looking at RT-PCR or through histology?
We did histology.
Sweet, thanks.
And Andrew, this unique profile that you're seeing in the neutrophils, is it naturally occurring or only find it when you add the zymosin and other sort of redirecting factors?
So I've only looked at it with the zymosin and the sterile inflammatory activating factors. So that has been Dr. Jerome in Dr. Segal's lab is looking at, what his project is looking at what makes them alternatively activated at the, can we generate these de novo without the presence of the zymosin? And that is a process that he is still working on. We know that that IL-4 is a very important factor in that process, but we haven't figured out the rest of that.
Okay. Let's see, we have any more questions? Anybody else want to pop in live here? I'm not seeing anything, Andrew. I don't know if you are seeing anything.
I appreciate it. I'm always, if people have questions or something comes up, they can always email me. I'm happy to, talk with people about any of the stuff that I talked about today.
Yeah. Well, today you're competing with the National Neurotrauma Society has their annual meeting right now.
I did not know that.
Yeah, and I'm going there next. So that's my next virtual place. I get to listen to myself give a talk, I guess. But yeah, thanks so much. And, really comprehensive, exciting work that you're doing, and look forward to seeing what you have in the future. And particularly as you do some more SCI and TBI type studies, it'll be exciting. So thanks everybody for joining us, and we'll be back again a month or two. And we'll put the word out for those lectures as well. So thanks again, and have a good day, everybody.
All right, bye.